Good morning, everyone. It is my pleasure to introduce Dr. Goldsworthy from USD, Keck School of Medicine. He is an associate professor of research in otolaryngology, head and neck surgery, and director of the Bionic Ear Lab. He received his Bachelor of Science in Physics from University of Kentucky and his PhD in Health Science and Technology from Harvard University and Massachusetts Institute of Technology. He is a cochlear implant user himself and is passionate about interplay of auditory experience and auditory perception. His research focuses on combining psychoacoustic, signal processing, and auditory rehabilitation with emphasis on cochlear implant technology. We are very excited to have you, and now welcome. We're looking forward to your talk. Thank you so much.
Yeah, thank you. Thank you for that introduction. So I'm gonna get started here, and if unless anybody has trouble like understanding me without my video on, I'm just gonna turn it off. I've been trying to have better posture, and I want to look up at my higher screen, and I don't want everybody looking down my nose. So I'm going to turn my video off if I can. Okay. And then I will get into it. Just want to make sure that the Zoom stuff is out of the way. Yeah, so thank you for having morning coffee with me, morning tea. And I'm going to be talking about the importance of stimulation timing for cochlear implants. Let me get in here. This is my lab. It's a fun lab because we're at the intersection of music perception and biomedical engineering. So we have people are very quantitative in the lab who are interested in signal processing and in designing and developing better signal processing for cochlear implants. And we have musicians and artists in the lab who kind of drive a lot of the exploratory questions about what are the limits of music perception for people who have cochlear implants. And so I'm kind of at the center of this and help translate the two languages the engineering speak and the musicians speak between the two types of students and trainees that come to my lab. And we study music perception in cochlear implants. That's a major part of what we do in the lab. And this is a nice visual metaphor for why listening with a cochlear implant to music is so difficult. Sort of the child with mittens on. It really comes down to pitch resolution or the lack of pitch resolution. Pitch resolution can be rather coarse with a cochlear implant. And because of that, it affects all other aspects of music perception, from timbre and tonality, even rhythm perception to some extent can be affected by pitch perception. So it's something that we're very interested in. And we also develop signal processing to try to improve cochlear implant outcomes. So what can we do to actually make the sound processing, taking sound and turning that into electrical stimulation to improve outcomes for cochlear implant users? So that's very much what we try to do in the lab. And before I get into cochlear implants too deeply, I want to just kind of preach to the choir, and I'm pretty sure I'm preaching to the choir here, that medical bionics are miraculous. And I want to say that beyond cochlear implants, but we're living in exciting times in more than one way. But one of the ways is that we're helping people walk, run, and dance again if they lose their leg. For example, this lady, she lost her leg in the Boston Marathon bombing. She was a dancer and a marathon runner, and she wants to do more than just walk on a leg. She wants to run and dance on it. And biomedical engineering is now at the point where that kind of functionality can be restored, not perfectly, but can be restored. Likewise, people who lose their arms can have their arms restored, and people who lose their ability to see or are born blind can have some degree of vision restored to them through biomedical devices. Also, disabilities and diseases are treated with deep brain stimulation, and spinal cord stimulation is used to alleviate pain. And I wanted to say all that because cochlear implants are an example of this, and a leading example. They're perhaps the most successful biomedical device for restoring a sense. So they are. And studying cochlear implants can inform the development of those other biomedical devices. So I wanted to mention that. And then I want to dive into how do cochlear implants work? What is the basic function? I know everybody here, let me see if I can get a laser pointer. I know everybody here is pretty familiar with it, but I just sort of a short summary. A cochlear implant is for somebody who is otherwise profoundly deaf in that ear. And, as you know, normally sound goes into the ear, vibrates the eardrum, the ossicle chain, down to the stapes, which vibrates back and forth. The acoustic energy of such is pushed into the cochlea, and inside the cochlea there are hair cells which pick up the vibration, very much like blades of seagrass that bend, and that cascade of action allows people to hear, excites the auditory nerve and allows people to hear. And if somebody has sensorineural hearing loss, so the cochlea, those hair cells are damaged, then a cochlear implant is a way of allowing that person to hear again. And the way that it works, as is that a electrode array is surgically implanted with as many as 22 electrodes, so individual stimulation sites, and as few as one stimulating site. And then these electrodes are going to directly electrically stimulate the auditory nerve. So truly miraculous. You can go from being deaf to being able to hear, like myself, quite well. And so the other parts of the cochlear implant is there has to be a microphone somewhere to pick up the sound energy, and that's most typically situated in a behind the ear processing unit. So there's a microphone, or to be precise, microphones. There's usually more than one microphone. There's usually two that fit in this behind the ear processing unit. And that's important because once you have two microphones, you actually can do some triangulation of where is the sound coming from. So you can build upon that some look to listen sound algorithms where you can emphasize whatever you're looking at. You can design different kinds of spatial hearing algorithms using multiple microphones. So the microphones pick up the sound and bring it to a computer chip. The computer chip, the job of the computer chip is to take that sound energy and convert it into an electrical stimulation pattern. And in my mind, that's where a lot of the magic of the cochlear implant comes into play, because it's not a solved problem. We don't know, we haven't developed or designed the optimal stimulation pattern so that you can, once you have that microphone signal, what is the math you need to do to figure out what these electrodes, how they should fire, given the constraints of the implant. So in my opinion, there's still a lot of work that can be done in terms of optimally developing sound processing algorithms designed to optimally stimulate the auditory nerve given whatever implant constraints that you have. So that's the cochlear implant in a nutshell. And now I want to make the point that timing in the auditory system is very important. And I like to say, and I'm pretty sure it's true, that the auditory system is the temporally most precise biological system that we know of. And I think that's best exhibited by your ability to hear where a sound is coming from in the environment. I'll get to that a little bit later. But I want to make the point that in the auditory system, there are special biological mechanisms for encoding precise timing. So, for example, if you look at the inner hair cells, the way that these are those blades of grass that bend in the cochlea when the acoustic waves go back and forth, these are coupled to the auditory nerve, and they need to, or they do, encode precise timing. And one of the mechanisms is that there's ribbon synapses that provide fast and sustained vesicle release across from the inner ear cell to the auditory nerve. And that promotes fast timing in the auditory system. Another example is when the auditory nerve goes to the next place that it goes to, which is the cochlear nucleus in the brainstem. There's special synapses, calyxes of health, shown here, where there's a very tight coupling of the information between the auditory nerve, seen here in yellow, and the a principal cell in the cochlear nucleus. And that it not only provides a very tight transmission of the timing of an action potential, it actually enhances the timing of an action potential. The timing is very important, as we can see biologically. And then I want to point to this nice paper by Golding and Ortel back in 2012, where they're talking about some of the precise timing mechanisms. So one of the points that they make is that when you look at the cochlea, let me roll back real quickly. When you look at the cochlea here, one of the things I should, of course, mention is that the sound energy, the low frequencies go all the way up to the apex, and the high frequencies selectively excite the base. And that actually creates a traveling wave delay. With the traveling wave delay, it takes low frequencies longer to get up here and eventually excite the nerve, compared to high frequencies which excite the nerve right away in the base. So there's this traveling wave delay. There's a specialized mechanism in terms of the dendritic arborization of the octopus cells in the cochlear nucleus that correct for that. So you can actually have synaptic growth that corrects for traveling wave delay, that makes the arrival of incoming action potentials time-aligned and highly synchronous. There's a similar structure in the MSO of the brainstem, which is responsible for integrating timing information between the left and right ear, so you can tell where sound is coming from in space. So you can use the timing differences across ears to tell where the sound is coming from in space. So there's several biological mechanisms that the end result is very precise timing in the auditory nerve in normal hearing. And so I wanna unpack that a little bit more. And so the way that I'm gonna unpack it is I'm showing you how the auditory nerve responds to a very simple sound here. I'm showing you here the output of a computational model of a healthy auditory nerve connected to all the parts in front of it. And I'm driving it with a pure tone, and I'm showing here the characteristic frequency of a nerve fiber. So you're thinking here, these are all nerve fibers on the y-axis, where I'm stimulating with a 500 hertz pure tone. And because I'm stimulating with a 500 hertz pure tone, the characteristic nerve fibers are selectively responding. Of course, that is tonotopy, or the frequency coding in the auditory system is the hallmark of the auditory system. And if I change that to a 1000 hertz tone, the place of excitation correspondingly changes. And so that's, again, tonotopy. But I also want you to see that on a cycle-by-cycle basis, the timing is phase locked to the incoming tone. If you're stimulating at 500 hertz, the auditory nerve will respond at 500 hertz. So there'll be action potentials clustered every 2 milliseconds. But if you go to 1000 hertz tone, then the auditory nerve is firing with clusters every 1 millisecond, which so synchronized with 1000 hertz. And so the normal hearing brain has access to very clear place of excitation cues, but also stimulation rate or stimulation timing as a clear cue for frequency or for pitch perception. And we don't really know how much the normal hearing brain uses these two pieces of information. We just know that it gets both pieces of information. Now, in contrast to that, the conventional cochlear implants generally do not use precise timing. So if I made a similar figure, but showing the stimulation pattern coming out of a typical cochlear implant for a 500 hertz tone and for a 1000 hertz tone, you see, oh, hey, the place of stimulation, the electrode that is firing changes. So it's doing a good job of conveying tonotopy, or as good of a job as possible if you only have 22 electrodes in this current spread, but it's doing a pretty good job of conveying that. However, there is no information in the stimulation timing. So the conventional, the most commonly used way of stimulating a cochlear implant just use a fixed rate stimulation, often 900 pulses per second. So if you play a 500 hertz tone, the implant stimulates at 900 hertz. If you play a 1000 hertz tone, the implant stimulates at 900 hertz. And so it's not using that timing information. There are exceptions, and in particular, Med-El Corporation has a fine structure processing algorithm that is trying to put that timing information back in. But still today, the most common conventional way of stimulating completely discards that timing information. Okay. So with that, cochlear implants generally use stimulation place rather effectively, but stimulation rate is not used to encode acoustic frequency. And this is one of the main messages that I want to get across, is that I think stimulation timing is important, and the field should try to move towards putting that timing information back into the stimulation strategy. Okay, so with that, how do cochlear implants typically stimulate? What is the most common way of stimulating? So the most common way for cochlear implants to work, the most common signal processing, is that sound comes in and is broken into a bank of bandpass filters, so kind of emulating the basic function of the cochlea. And so it's broken up into high frequencies down to low frequency processing channels. The output of that, the temporal envelope, is extracted. And this is the part where the faster timing information, which we refer to as the temporal fine structure, the fine timing information, is discarded. And there's historical reasons why this algorithm was developed, and I can kind of go into that. But the key point is this is where it's discarded. And then that temporal envelope information is used to modulate pulses. So let me try to give you a little bit of intuition for how this translates for a specific sound. So I'm going to walk into the engineering side of things just a little bit. And so the way that I'm going to do that is I'm showing the word choice at the output of bandpass filters. If you take the word choice, then there's a high frequency burst at the beginning, the ch sound right here. And that high frequency, when you say ch, the tongue kind of comes to the front of the mouth, and so it's making a narrow cavity. Because it's a narrow cavity, there's a lot of high frequency resonances. It's an unvoiced sound. Your vocal folds are not vibrating when you make the ch sound. And so you get this high frequency burst of information up here, and then that leads into the vowel. So the vocalic portion here, the oi, is for someone with a trained eye at this, you can actually see the resonant frequencies or the formant frequencies associated with the vowel oi. And oi is a diphthong because it kind of goes from one vowel, more of an O, to an E, oi. And when it does that, you can see the movement of the second formant and a little bit of the first formant here. Not only that, but there's clear periodicity here. If you kind of zoom in here, and I think I zoom in the next slide, maybe not. So there's clear periodicity here where you can see an up and down peakiness. That actually corresponds to the fundamental frequency of voicing, because when you make a vowel sound, that is a voice sound. Your vocal folds are opening and closing in a periodic manner. And so you see some of the voice pitch information here. And then at the end, you got another sound, so the tongue comes even further to the front. It's a very much a high frequency noise burst. So that's what this word looks like at that point of going through a bandpass filter. It's got a lot of information in it. It's got a lot of place of excitation information. There's multiple channels. It's got really exquisite timing information if we zoom in. The next stage of conventional cochlear implant signal processing is to get rid of the timing information, the fast timing information. You still get the temporal envelope, and in the temporal envelope, you still see some of the periodicity associated with the fundamental frequency of voicing for the vowel here. You get the main components. There's a burst of energy at the high frequencies here. There's still the formant frequency information, and there's a burst of energy here at the end. And then the next stage is to take this envelope and to modulate a pulse train. And at this stage, the timing information, depending on the underlying pulse rate, is even further degraded. Nevertheless, it's got a lot going for it. It's got this burst of information up here for the cha. It's got some of the periodicity, although awfully watered down at this point. You can still see the formant frequencies here, and you can see the part here. Okay, so what is the point of all this? The point is that there's a lot of speech information that is captured by this way of doing signal processing, which aligns perfectly well with the fact that most cochlear implant users, even using this strategy, can understand speech in quiet quite well. Speech is a very redundant signal, and this captures the key pieces of information despite completely discarding a lot of the fast timing information, the temporal fine structure, or the fine timing. And so it makes sense that today, even with this being the leading strategy, and with about 1 million people having received cochlear implants, that they're quite able to understand spoken speech in quiet situations without needing to lip read. However, it's clear that the main problems with cochlear implants are music appreciation and speech comprehension in noisy situations. And I should also say there's their ability for bilateral cochlear implant users, so cochlear implant users who have an implant in both the left and right ear, their ability to determine where sound is in the environment is also degraded. So these are sort of the main problems with cochlear implants today, and many people, including myself, hypothesize that precise timing is the key feature that is limiting these kind of outcomes. So I want to make the point now that cochlear implant users can learn to use precise stimulation timing. And so let me just dive right into this. So clinical devices, the point that I just made, most clinical devices do not provide precise stimulation timing. But research has shown that precise timing can be transmitted from the implant to the nerve. So there's been a host of animal studies, and there's been a host of electropsychophysical studies where research tools are used to provide precise timing to either cochlear implant users or to animals, and to see how well that timing information is transmitted. And the animal studies are particularly impressive. So what I'm showing here is that so let me set this up a little bit. So there have been many studies where a cat or a rabbit has been given a cochlear implant, and that implant has been used to electrically stimulate, and here using a sinusoidal electrical current, electrically stimulate the auditory nerve, and then the scientist is measuring the auditory nerve activity. So these are action potentials in the lab animal. And the point that I'm making here is that the action potentials are exquisitely phase locked to the stimulus. So the action potentials line up with a particular phase. And so the key question is, how fast can you stimulate and still have these action potentials phase lock to the stimulus? Because that's going to really show you what is the potential limit, biological limit of the system. And it's exquisite. It's really the ability of the nerve extends at least up to 3 kilohertz. So what I'm showing here is the synchrony, or the phase locking ability, the synchronization index. So we're increasing the stimulation rate or frequency here, and then looking at what point can the auditory nerve no longer keep up. And so the open circles here is acoustic stimulation, so playing a pure tone into a cat's ear while measuring the auditory nerve activity. And then these two are electric stimuli, so providing a cochlear implant to the cat, electrically stimulating and seeing how well the nerve can phase lock. And not only does it phase lock as well as normal, it's actually better. There's a higher degree of synchrony, and that's because you're bypassing some of the sluggishness of the transduction process. So from a purely biological standpoint, timing of stimulation can be transmitted to a lab animal with a high degree of fidelity. So this is very promising. And so the next question is, can stimulation timing improve frequency discrimination? And then related to that is, how good is frequency discrimination in cochlear implant users? Kind of returning to that earlier picture of the boy with the mittens on playing the piano, limiting frequency resolution. Well, how good is it, or how bad is it? And so I want to put that in perspective here. Of course, everybody here is very familiar with pure tone audiometry. How well can somebody detect a sinusoid or a pure tone? You know, it's routinely used in the audiology clinic to probe hearing. That's not quite what I'm showing you here. I'm showing pure tone frequency discrimination. So that same pure tone is turned up to be loud and clear, usually about 65 dB SBL, so you can clearly hear the tone, and then the frequency of the tone is adaptively adjusted to determine the just noticeable difference. So how well can people with normal hearing tell apart two different tone frequencies? That's what this is showing. And this yellow curve is normal hearing, as well described by Christophe Michel and his colleagues back in 2012. It was a meta-study, so he's looking at several different studies, but this is how good frequency resolution is in normal hearing. I have a dotted line here showing one semitone, which is, you know, musically speaking, an important degree. So this is the just noticeable difference in a percent difference. So one semitone is about a 6% difference here. And so you can see that normal hearing individuals typically have frequency resolution much better than a semitone, you know, down a quarter to a 16th of a semitone. And that ability to hear microtones really opens up the rich tapestry of tonality that is present in music. Now, these are cochlear implant users measured in my lab, and there's several things that I want to point out here. Optimist that I am, I want to lead off with the fact that many cochlear implant users particularly in that mid-frequency range, are encroaching upon the normal hearing discrimination threshold. So they're able to use place of excitation cues to hear very small differences in frequency. However, really got to point out the broad range of outcomes in cochlear implant users, with some requiring 4 semitones or more to be able to hear a frequency difference. The average tends to be a little bit better than a semitone for mid frequencies, but worse for low frequencies. Just to kind of scale you, middle C is around here, and then treble and bass clefs over here. Or another way to scale it, typical male voice pitch would be about 110, a typical woman voice pitch would be 220, and a child would be around 440. And so you can see this characteristically bad performance, particularly for the low frequencies, is an important thing, because, you know, ecologically, I want to be able to hear your voice pitch more than anything else. And so I want to be able to hear that. And so being able to bring these down and provide better resolution is important to me. And if there's any frequency or the timing information is going to be particularly helpful, it's for the low frequencies. And so there's a lot to take from this kind of analysis. So then many people in the field, myself included, we asked the question, well, what if we bypass clinical processing? I just kind of disparaged clinical processing earlier, and I said that it discards timing information. What if we use research tools to bypass clinical processing and provide precise timing in laboratory studies? And we can do that. Well, you know, there's research hardware that can be used. It's provided by the implant companies that allow stimulation timing to be provided with microsecond and in many cases sub-microsecond precision. So very precise timing can be provided in lab studies. And studies that have examined pitch perception and sound localization with precise timing cues. So there's been decades of research doing this. So for example, the stimulation electrodes can be held constant, so there's no place cue being provided, just the timing cue. I'll give you an example here, just showing where there's like 5 electrodes active, and then the same 5 electrodes active over here, but the stimulation rate, that is the speed that the stimulation increases from here to here. And cochlear implant users, when they hear that, will typically say that this is higher in pitch. So they're hearing that as a pitch cue. However, historically, even when these kind of research devices are used to provide precise timing, cochlear implant users don't seem that good at using this information. To kind of bring you back to this scale over here, so this is the same sort of normal hearing frequency resolution. And this is a study by Fang Gang Zhang back in 2002, where they measured stimulation rate, so purely timing pitch perception in cochlear implant users. You can see they're not that good at it. In the very low frequencies, it's about a 10% difference, between 10 to 20%, so about between 2 to 4 semitones. And then it gets worse, and it tends to roll off rather steeply around 300 hertz, which has provoked many people in the field to say that there's a 300 hertz limit, a timing limit, above which cochlear implant users aren't good at using timing. Which, if that is true, is really disappointing, because you see that the auditory nerve can phase lock at least up to 3,000. So if there is a 300 hertz limit, then that's really you know, an order of magnitude loss of information for cochlear implant users. However, I have to make the point that most of these lab studies don't provide extensive familiarization or training with the new cue. And so just, please, if there's anything to take from this presentation. This is it. And clinical cochlear implant, the typical way of stimulating, does not provide fine timing. And so if we take somebody into the lab and study how well they can use fine timing in the lab, they're hearing it for the first time with this level of precision. And so we shouldn't expect them to be that good at that. If there's anything we learn from cochlear implant science, it's the brain is plastic. The more you listen, the better you hear. The brain needs time to make use of this new information. So we push that a lot in my lab. We try to do these kind of carefully controlled timing experiments, but provide increasingly more familiarization, more training with the cue. And so these results, which has been replicated many times, have led many people to believe there's only a 300 hertz limit for place pitch. However, those studies do not provide participants with training for the new cues. The central hypothesis in my lab is that if you give cochlear implant users this information, they will learn how to use it. The key point is to provide these timing cues in a clear and consistent manner. And so the general methods in my lab, we use research hardware to bypass clinical sound processing to provide precise stimulation timing. We measure behavioral thresholds and interoral timing differences, discrimination, and we use extended psychophysical training up to about 30 hours over the course of multiple weeks. And it's exciting because when we do that, so this is before training, and I would say that compared to the previous study here, even our baseline measure extends a little bit further because it's still more familiarization to the cues that they're listening to. But then after training, there's this large improvement. And that's very exciting because it's sort of showing that this can start encroaching upon that normal hearing region. So this is a much more potential cue for pitch perception than previously thought. So the effect of training is large, and it is possible that further training will lead to further improvements. Again, we're doing this with research hardware. They're not walking around with this information yet on a daily basis, but with the research hardware provided in a clear and consistent manner, we see that it seems to be well fit or well described by linear improvement over time, and we don't know how much longer this will sustain. And so that's one of the pressing questions of the lab. And so recent finding from the lab is that pitch discrimination is better with combined use of place and rate of stimulation. Pitch discrimination is better with variable pulse rates than with modulated pulse trains. And pitch discrimination is more robust to nearby electrical interference with variable pulse rates with modulated pulse trains. And this is, so I want to give you a little bit of insight to what I mean by this, and then just kind of like point to the papers that are associated with it. And so, again, a sense of pitch can be provided by the place of stimulation, that is, which electrodes you stimulate produce different pitch, with the ones that are more deeply inserted providing a lower pitch. In a similar way, stimulation rate by itself can produce a sense of pitch, with the faster stimulation rates being heard as higher in pitch. But then you can combine the two cues, and if you co-vary the place and rate of electrical stimulation, you can get an even better sense of pitch. And that's seen over here where place pitch is in blue. There's again discrimination, so just noticeable difference. Same thing over here in semitones, one semitone right here. And this is a measure of maybe 6 or 8 subjects that we tested for this paper. And it seems like the combined use of place and rate is better than either cue alone. And that's important because clinical sound processing does not stimulate this way. It does not co-vary the place and rate of stimulation. So this is a cue that is not currently provided to cochlear implant users, and we should provide it in the hopes of giving them better pitch perception. Likewise, most existing clinical strategies modulate a fixed rate pulse train here rather than using a variable pulse rate as shown here. So here the pitch or the fundamental frequency is the distance between this peak and this peak. It's kind of conveyed by amplitude modulation of this electrical pulse train. And then here it's conveyed by a variable rate pulse train. And the main point here is that the variable rate pulse train provides better discrimination of pitch changes. So that's the main point here. And then this dog is a quick way for us to catch our breath and switching topics a little bit to sound localization, because we've recently started doing sound localization experiments in the lab for telling where a sound is coming from. There are two primary cues are the level differences and the timing differences between ears. So if a sound is coming from over here, of course it's going to get to the right ear first, so there's going to be a timing difference. And there's also going to be a level difference. It's going to be louder in the right ear. There's also a spectral notch difference, but I'm not going to go into that one. And I just want to say that cochlear implants do a decent job of conveying these level differences, but because bilateral processors are not coordinated or synchronized. If somebody has two processors on, the left and right processors are not talking to each other in any way, but they're now at the point technologically where they can. We can use Bluetooth and other wireless technology to communicate the stimulation that's delivered by the left and right implant. We're getting to the point where we can do precise timing. But I have the same hypothesis, but with this different perception. The hypothesis is that cochlear implant users will improve their interoral timing discrimination, that is their ability to use timing differences across the ears to make, if they're provided this information in a clear and consistent manner. So again, let's take a look at how well people with normal hearing can hear the interoral timing differences. This is a summary of such from normal hearing listeners for pure tones. Is exquisite with this is the discrimination threshold or just noticeable difference in microseconds. And the best performance can get down to 10 microseconds. So being able to hear a pure tone that differs only by 10 microseconds when presented to the left and right ear. And this is normal hearing performance, which falls off rapidly above about 1500 hertz or so. And so that's the holy grail that we would like to provide to cochlear implant users. And we have no idea how good it's going to get for cochlear implant users, because we have not yet coordinated nor synchronized their clinical sound processors. So that's yet to come. In the meantime, there have been decades of research using laboratory studies, again, without training, but providing highly synchronized left and right stimulation across ears to see how well cochlear implant users can hear this information. This is a summary figure of that. Cochlear implant users tend to be all over the place, but in general, much worse than we see in normal hearing, all over the place. Shouldn't be surprising. Many of these individuals have been deaf for a long time. Their implants are not synchronized. They're not getting that information. So the biological state has kind of fallen into an impoverished state. But in general, the region here is not nearly as good as this holy grail here. And so I asked the question is, well, is it learnable? Is it something that we can train in the same way as with the rate pitch studies? We provide this information in a clear and consistent manner. We have them train on it for about 30 hours, and we see if they get better. And this is the pre-assessment, so sort of middle of the range for your average bilateral cochlear implant user. And then this is post-assessment, so after 30 hours of training. And we don't know how much better it's going to get with further training, really getting to that edge of we have to now provide this on a daily clinical basis to see if daily listening with structured engagement activities can improve this performance and bring this down. And until we do that, we don't know what the true limit of this timing information is. And I just wanted to show here another slide, which is showing that output for the CIUs in the lab, and this ability of normal hearing listeners to do something similar with acoustic pulse trains. So it's quite possible that the true limit for cochlear implant users is not this yellow curve, but is this red curve. And if that was true, it would still be a great benefit for cochlear implant users, but I hope it's not. I hope they can get to this yellow curve at some point. But I just wanted to put this out there that this is quite possible that cochlear implant users, even in the best case with training and synchronization, will asymptote at this red line, which is acoustic pulse trains rather than pure tone pitch perception. And I'm happy to unpack that too. But presently, we do not know how much further learning will continue with further training. This is just training over time for 3 of the subjects. I need to update this we have 8 subjects who have completed the training so far. It tends to be pretty linear, and it doesn't show clear signs of asymptoting, except for this subject here, which, he's down at 30 microseconds, and so he's quite good at the task. And if that is his true limit, I'm sure that every other bilateral cochlear implant user would gladly take that. But let me wrap up. So in summary, stimulation timing is important for both pitch perception and sound localization in cochlear implant users. Stimulation rate and intraoral timing discrimination can be improved with psychophysical training. Most clinical stimulation strategies discard this fine timing information, and my lab focuses on providing stimulation timing in a clear and consistent manner. I want to acknowledge funding support, NIH and R01 on encoding temporal fine structure for cochlear implants. That is primarily what I talked about today, and then an R01 on music appreciation after cochlear implantation, which I barely touched upon today, but I'm happy to follow up on music appreciation after cochlear implantation in depth with anybody who wants to follow up with me on that. And I want to thank the Hearing and Communication Neuroscience program, like many other training programs at NIH right now. It's in danger. Hopefully it'll continue. But it has been a strong supporter of research in my lab, supporting postdocs and PhD students for the past 10 years. So I'm very thankful for that. On that note, if there's anybody who's interested in doing a postdoc or a PhD in the hearing and communication neurosciences, just reach out to me. And again, my lab, fantastic people, and I'm very thankful for them. And thank you for listening.